In this video, we will be talking about enzymes. Chemical reactions are slow taking place, so in order to speed up these reactions, our bodies use specific proteins called enzymes. The use of enzymes are necessary for our bodies to function well. For example, imagine feeling tired and in need of energy, so you eat a snack to get that energy, but that energy doesn't come until 5 hours later. This is what our bodies would be like without the use of enzymes. Now we will examine the 4 major characteristics of enzymes. The first characteristic of an enzyme is that it is reusable. Enzymes have an active site where substrates, also called reactant molecules, bind in order for a chemical reaction to take place. When a substrate binds to the active site, it is broken down and is turned into new products. Once the products leave the enzyme, it is free to be used again. The second characteristic of an enzyme is that it is a catalyst. A catalyst is an agent that speeds up chemical reactions. Enzymes speed up chemical reactions primarily by lowering the activation energy. Activation energy is like a shield that 2 molecules put up. Enzymes break down this shield to speed up chemical reactions. Enzymes use 3 different techniques to lower activation energy. The first way is by binding to the reactant to loosen the bonds between the substrates. The second way is by orienting the reactions closer together so they can actually bond, and the third way is by temporarily changing the local environment of the reaction. The third characteristic of an enzyme is that it is highly specific. The active site on an enzyme is built specifically for a specific substrate molecule. One way to think about it, an enzyme is like a combination lock. Only the right combination will unlock the lock. In the same way, only the right substrate can bind to the enzyme for a chemical reaction to take place. The fourth characteristic of an enzyme is that it is very temperamental, meaning it is very sensitive to its environment. Different conditions can change enzymatic activity. Differences in substrate concentration, the presence of inhibitors, or change in temperatures can affect enzymatic activity. We saw how substrate concentration changed enzymatic activity in lab. We saw that the higher substrate concentration there was, the greater the amount of enzymatic activity. However, at lower substrate concentration, we saw that enzymatic activity was also low. This occurs because there are many enzymes that are inactive due to the lack of substrates. With dealing with temperature, we also saw that enzymatic activity was low at extreme temperatures. This is because enzymes have a narrow range in which they will perform optimally. Lastly, we saw how the presence of inhibitors can affect enzymatic activity. There are 2 types of inhibitors. There is competitive inhibitors and non-competitive inhibitors. Although both of these inhibitors work differently, they both lower enzymatic activity. Competitive inhibitors bind to the active site on the enzymes and block the substrates from bonding. In non-competitive inhibitors, they bind to another part of the enzyme called the allosteric site. When bonded to this site, this changes the structure of the enzyme and therefore does not let the substrates connect to the active site. So in this video, we have talked about how enzymes work and how they are reusable, how they are catalysts, specific, and temperamental.